Okay, thank you very much. So first and foremost, I would like to acknowledge First and foremost, I would like to acknowledge the traditional custodians of the land to which this research was done and pay my respects to the elders past and present. So my presentation today, as it has been mentioned, is about the interaction of the Australian tapered fruit flies with the twisted wing endoparasitoid Dipterophagus dacey and the endosymbiont Wolbachia. And this work was published in Environmental Biology, just in case you need further information regarding this work. So the Queensland fruit fly is Australia's major tapered pest, and many control methods have been put in place to control these pests. One of the methods include the sterile insect technique, or SIT, that involves sterilization by irradiation, release of the irradiated flies for population suppression. Alternatively, the incompatible insect technique that uses Wolbachia for sterilization has shown a lot of, is really promising in the control of insect pests and disease vectors. So what is Wolbachia, you may ask yourself? So these are intracellular endosymbionts that affect approximately 50% of insect species. They are primarily transmitted from mother to offspring. However, there's occasional horizontal transmission of Wolbachia from one host species to another, and it can lead to either the infection being established in the new population or can just remain as transient infections. So Wolbachia can manipulate host reproduction, for instance, cytoplasmic incompatibility, or CI. So CI occurs when a male that is infected with Wolbachia mates with a female that is not infected with Wolbachia or is carrying a different strain of Wolbachia. So this ability for Wolbachia to cause CI has been explored for this insect pest and disease vector control. So however, some insects and disease vectors that are actually are not naturally infected by Wolbachia. And in such instances, Wolbachia has been artificially transferred by embryonic microinjection into this insect species. However, the key thing for IIT is to identify a suitable Wolbachia strain. So my project started with the aim of applying this principle of IIT in Bactrocera triani. So what we did is we performed microinjection of W. male Wolbachia from Drosophila melanogaster into Bactrocera triani, followed by collection of pupae and then monitoring of adult emergence. And this picture just shows the step-by-step process of what we did. So with this experiment, we were able to inject approximately 28,000 Bactrocera triani embryos. And we managed to get about 8.7% females infected with Wolbachia. And these females were isofemale. And we only managed to have one female being infected, one Wolbachia infected female transmitting this infection to its G1. And unfortunately, this infection was lost at G1 stage. So, well, we had to go back to the drawing board. So a previous study by Jane Morrow from our group identified two Wolbachia strains that were then designated as ST285 and ST289 in 7 out of 24 Australian tapered fruit fly species. So these Wolbachia strains had very unusual characteristics. For instance, they occurred in very low prevalences, and identical Wolbachia strains were detected in several tapered species. Furthermore, these infections are only restricted to the northern part of Australia, and they had no link to a mitochondrial haplotype. And last but not the least is they had unknown host effects. So these unusual Wolbachia infections have not only been reported in tapered fruit flies, but also in other insects. And this slide just shows a snapshot of these other studies that have observed these very unusual Wolbachia infections. So the big question is this unusual Wolbachia due to horizontal transmission, or is it due to the detection of Wolbachia in a concealed host, for instance, parasitoids, and the inadvertent assignment of this infection to a wrong host? So parasitoids attack a wide range of insect orders, and endoparasitoid wasps, for instance, are common in fruit flies, and they've been shown to have Wolbachia. And in addition to that, also, a strepsipteran endoparasitoid has called Dipterophagus daisy has been described and recorded in 19 Australian tapered fruit flies. And in other insects, strepsipterans have been shown to also carry Wolbachia. So our main questions were that we wanted to really understand what maintains these strange Wolbachia infections in fruit flies. And do fruit flies have a concealed endoparasitoid that could be a potential host of Wolbachia? And if yes, is there a link between Wolbachia and the presence of this endoparasitoid in fruit flies? So to do this, we collected samples from Central and Northern Queensland. And this map shows us the regions in which we collected our samples, including the number of samples that were collected from each location. And these samples spanned, were male tephetids from seven species that are just listed on the slide. So these collections were facilitated with the help of Jane Royer and Sharon from QDAF. So for the first part of our experiment is that we wanted to characterize these unusual Wolbachia infections that were detected in tephritid fruit flies. So to do this, we used 13 Wolbachia positive fruit fly DNA extracts. And with these sequences, we used C. capitata mitogenome to be able to filter. And first of all, to just check the quality of our sequences. We just wanted to see the fly mitogenome. So after filtering, it was followed by assembly by read mapping. So we also checked Wolbachia, and to do this, we used the W-mail genome, followed by assembly and read mapping. So results from this first part of the experiment is that yes, our libraries had the expected fly mitogenome. However, in addition to the expected fly mitogenome is that there was an additional mitogenome belonging to another insect in 6 of our libraries, and the remaining had traces. So our table here just gives us a summary of the 13 samples that we did whole genome sequencing. So preliminary identification of this additional mitogenome showed that it belonged to a strepsipteran, or else called a twisted wing parasite. So we wanted to understand more of, why do we have this additional mitogenome in our libraries? So what we did is we used the Mengenilla australiensis strepsipteran mitogenome as a reference for extraction and filtering of these strepsipteran sequences, and then followed by assembly and read mapping. So a small background about Strepsiptera. So it comprises of approximately 630 species, and they parasitize 35 insect families and 7 insect orders, as listed here. And they have a very unique life cycle and an extreme sexual dimorphism. So the picture here is an example of a life cycle of Xenosvesparum strepsipteran. And the females are fully endoparasitic in their host, while the males are free living, and they are endoparasitic and they emerge after the 4th instar larvae. So for the second step is that now we have this additional mitogenome. What is it doing there? So we wanted to understand and to investigate if there was a link between Wolbachia and the presence of this strepsipteran mitogenome or strepsipteran. So to do this, we designed primers that were specific to Strepsiptera CO1 gene. And we used 64 fruit fly DNA extract to screen for both Strepsiptera and Wolbachia. And for confirmation, we did Sanger sequencing of both Wolbachia and Strepsiptera. So results from this is that our Sanger sequences confirm that yes, the sequences that we found in our whole genome sequences belong to Dipterophagus daisy. And the samples that were negative for Wolbachia were also negative for D. daisy, suggesting a link between the two. So our heat map just shows us the link between D. daisy positivity and Wolbachia positivity in our fruit fly samples. So with the Sanger sequences that we had, and also the whole genome sequences that we had, we did phylogenetic analysis to just determine the phylogenetic placement of D. daisy in the Strepsiptera phylogenetic tree. And on the slide is a Bayesian phylogenetic tree. And we found that D. daisy belongs to Halictophagidae family of Strepsiptera. So moving along, we also wanted to know now, yes, we have Strepsiptera in our samples. What is their titer? And we wanted to also see the tissue localization of the two Wolbachia, now designated as WDC1 and WDC2. So to do this, we used 23 individuals of 8 tephridids, so that were visibly parasitized, or we can call it stylopies at this point by DDC. So we performed dissection followed by qPCR to detect the presence of both WDC1 and WDC2. So this very interesting or bizarre image here is a parasitized fruit fly abdomen with this red arrow pointing to the DDC male cephalotheca, and the yellow one is the female cephalothorax. So, and B is DDC male pupa and male adults. So our qPCR results show that yes, the two Wolbachia strains occurred at very high titers in Dipterophagous daisy relative in the DDC tissues relative to the fruit fly tissues. And also the fruit fly tissues that were parasitized were also Wolbachia infected. So, and this actually confirmed that DDC is actually the true Wolbachia host. So additional characteristics that we observed in the DDC parasitized flies is that there was superparasitism of up to 6 DDC male and female adults per fly. And in addition to that is that the aedegas in the parasitized male were really unusually positioned compared to unparasitized males. So finally, we now know that we have this endoparasitoid or DDC in our fruit flies. So what is the prevalence of this DDC in our flies? So to do this, we use 685 flies collected from 14 localities in Queensland, and the table just shows the summary of these regions. And these samples are fruit fly males comprising 7 species. And the map shows us a summary of the prevalence of DDC in the regions. So to conclude is that unfortunately, our microinjection did not result in a Wolbachia infected laboratory population. And so with our screening, we found that Wolbachia infects DDC and can only be detected in flies that are parasitized. And parasitization with Strepsiptera is not always visible, and which means that all the previous detections of Wolbachia in Australian tephridids are due to parasitization by DDC. And the tephridids in Australia may not have Wolbachia. So also is that the detection of this unusual Wolbachia in other insect taxa may also be due to undetected Strepsipterans. And the elongated aedegas in the stylopied fruit flies could also be due to parasitization. And last but not the least is that we demonstrated the possibility of obtaining both endoparasitoid and host sequences from the same library. So lastly, I would like to acknowledge my very supportive supervisors, Marcus Rigler and Jane Morrow, and our collaborators from QDAF, Jane Royer, Shannon Close, Geraldine Kellam, Zung, and every other summer scholar who helped in this study, and the Rigler group in general, not forgetting the funding agencies and our university. Thank you very much.
Thank you so much, Sharon. So we don't have any time for questions. If you do have any questions for Sharon, please direct them to her in the chat and she can address them in a direct message afterwards.